I'm Evan Balaban, and my colleagues and I would like to tell you about our research on brain activity in embryos. We did this project for two basic reasons. First of all, we were very interested in seeing when a young organism's brain turns on for the very first time. The second reason we were interested in doing this study is that we wanted to develop an experimental method for relating features of prenatal brain activity to later brain development and behavior. We knew from previous work that all embryos show a lot of so-called spontaneous movements, seen here in human fetuses recorded by ultrasound sonography. We therefore developed a way of studying our chicken embryos in an environment where they would be as undisturbed as possible and where we could make non-invasive recordings of their heartbeat, movements, and any sounds they produced.
Positron emission tomography is an imaging technique that allows us to record the brain activity of the embryos without disturbing them. We also did a CT imaging of the embryos to age them according to their bone development. As they become older, their bone becomes larger and more dense. We carefully take an egg from the incubator, gently open a hole in the top. We deposit a dose of our tracer substance on top of the breathing membrane where it gets absorbed into the bloodstream. Next, the egg is placed in a heated, soundproof chamber on top of a device that records heartbeats and movements from vibrations on the egg shell. We can play sounds to try to wake up the embryo. During the next 20 minutes, the tracer substance is taken up by the brain cells according to how active they are. After this, we are ready to image the embryo. The egg is placed on the scanner bed, connected to the anesthesia system to prevent body movements, and advanced inside the scanner. First, an X-ray CT scan is taken, and then the scanner bed automatically advances the egg to the PET section of the machine, where a PET scan is performed. Once the scan is over, the egg is returned to the incubator. We could age the embryos because of the big difference in volume of bones in the head and neck between the younger and the older ones, as seen in these three-dimensional reconstructions.
There are the recent advances in PET, high-resolution scanning technology, what have allowed us to perform these quantitative studies of the brain activation in embryos. This technology was unthinkable a few years ago, and that's the reason why there was almost no previous results, and that's the reason why we didn't know what to expect from our experiments, actually. This first image corresponds to what we call the low activation state. We can see how the activation now also appears in the forebrain and in the cerebellum. In this third set, we can see how the increase of activation in the forebrain and the cerebellum is clearly visible. In this fourth set, we can see how the whole brain of the animal appears very active.
To see if embryo brains can wake up, we played sounds that are very meaningful to them after hatching, or sounds we created that were acoustically similar but meaningless. We found that embryos hearing the meaningful chicken sounds had more higher brain activation than animals that heard the meaningless sounds. These latter embryos had higher brain activity patterns very similar to embryos kept in silence. We also found that the intensity of activation in the brainstem areas of embryos who were played the chicken sound was strongly related to the intensity of activation in their higher brain areas, a relationship that we also saw in awake animals after birth. Only chicken-stimulated embryos had correlated activations that were as intense as awake post-hatching birds. In other words, waking-like brain function. So one of the questions that this research didn't answer is whether waking the brain up early is a good thing, a bad thing, or an indifferent thing for later brain development. This is a question that only future research is going to be able to answer.